So let's talk about the adenylyl cyclase system. As you already know, there are systems that are responsible for the transmission of hormonal signals from the outside to the inside of the cell. So those signals which come from hydrophilic substances or neurotransmitters or hormones, these ones require receptors to effect cellular response because they cannot easily pass through the lipid bilayer. Those ones which are lipid soluble, for example steroids, these ones don't require membrane receptors to effect cellular response. That means they can easily pass through the lipid bilayer into the cell. That means they have intracellular receptors. But those which are hydrophilic, for example, you already know acetylcholine, for example, or epinephrine, norepinephrine, for example, these ones require receptors, membrane receptors, because they cannot easily pass through the lipid bilayer to effect cellular response. And one of these systems which are responsible for the transmission of the hormonal signal from the outside to the inside is called Adenylyl Cyclase System. What are the components of the Adenylyl Cyclase System? So as you already know, the cell is a lipid bilayer, right? So it's represented by these two lines. Now, one of the most important components of the adenylyl cyclase system is the membrane receptor. Every system which you already know, hormones or neurotransmitters, these require receptors to bind so that they can effect cellular response in a series of steps. So we have the membrane receptor. That means the receptor is on the cell membrane. And this receptor is called G-protein coupled receptor. G-protein coupled receptor. So why is it called G-protein coupled receptor? It's called G-protein coupled receptor because attached to it here is a G-protein. And this G-protein is made up of 3 subunits. 3 different subunits. These are alpha subunit, beta subunit, and gamma subunit. So it is said to be a heterotrimer, meaning it consists of 3 different subunits. So that's why this receptor is called G-protein coupled. That means attached to it is called, that's what we call it, G-protein coupled receptor. So what happens in the adenylyl cyclase system is that we're going to have a signal. This signal can either be a natural hormonal signal, for example, epinephrine or norepinephrine. And we can also have drugs, I mean drugs which mimic the natural neurotransmitters. Is going to bind to this. So we are going to have the signal binding to the G-protein coupled receptor, which is attached to a G-protein that has alpha, beta, and gamma subunits. The binding of the signal to the G-protein coupled receptor is going to lead to the phosphorylation of the alpha subunit of the G-protein. So we're having G-protein here. The binding of this signal, it can be a drug or a natural neurotransmitter, is going to bind to the receptor and is going to cause the inactive form. So that means the G-protein in its inactive form is in the form of GDP. So remember, this one naturally is a stimulatory G-protein. That's what we represent as GS protein. The binding of the signal is going to lead to the phosphorylation of this into G-alpha, GS-alpha, GTP. So the GS-alpha, GTP that has been formed is going to go to what we call adenylyl cyclase. As the name implies, the system itself is called adenylyl cyclase, but it is almost one of the most important parts of the system. The function of adenylyl cyclase is to convert very important ATP. So we have AC, adenylyl cyclase, is to convert. But before converting, what's going to happen? The G alpha GTP that were formed is going to activate. So it has a positive effect on the enzyme adenylyl cyclase, which is going to lead to the conversion of ATP into cyclic adenosine monophosphate. So that's why it is called adenylyl cyclase, meaning it's going to convert the adenosine triphosphate into its cyclic form, which is called cyclic adenosine monophosphate. What's going to happen next after this? Very important to note is that the cAMP that has been formed here is called a second messenger in this system. Most systems have their own second messengers. So for example, especially adenylyl cyclase has its own second messenger system, also called phospholipase C system, which also has its own second messengers. But for adenylyl cyclase system, the second messenger that we have is cyclic adenosine monophosphate. So this is going to take the signal and then it's going to affect the other enzymes. So the most important thing you need to know is that the cAMP that has been formed is going to bind to what we call protein kinase A. So protein kinase A, as the name implies, is an enzyme, right? It's a kinase. But in its inactive form, it doesn't have cAMP. So what is going to happen? So let's represent protein kinase A as this. Protein kinase A has this site on it. So let's say these sites, right? I'm sorry. So let's represent it in a better way. So let's give it this. So we have the regulatory site. So regulatory site, two regulatory sites and two catalytic sites of protein kinase A. What's going to happen is that the cAMP molecules that have been formed, so we have the 2R, so these are regulatory sites, and these are catalytic sites. So this one is inactive form of protein kinase A. So after the cAMP is attached to it, what's going to happen? The cAMP molecules here that we have are going to bind to it, to the allosteric site or regulatory site. So from the inactive form without cAMP is going to form the active form with the cAMP molecules, I mean 4 molecules of cyclic adenosine monophosphate. So it's going to make the protein kinase A active. So the process by which protein kinase A is converted into its active form with the help of cAMP is what we call allosteric activation, because cAMP is going to attach to the regulatory site of protein kinase A. The regulatory sites are also called allosteric sites. That's why we call the process allosteric activation. Finally, what's going to happen is that the protein kinase A that has been activated is going to lead to the phosphorylation of proteins, protein phosphorylation. So you are going to have a protein being phosphorylated. But remember, proteins are made up of amino acids. So you have polypeptides, right? And polypeptides are having amino acids. So which part of the amino protein is going to be phosphorylated? So we are going to phosphorylate the part of the protein or the polypeptide that has amino acids that contain OH group. So that means it can either be tyrosine, it can either be serine, or it can be threonine. So here we are going to lead to phosphorylated protein. So I mean a protein that is phosphorylated. I mean phosphate attached to it. So this is finally going to lead to a cellular response. So this is the function of protein kinase A. Very important thing to note. One may ask, is this one going to be forever? That after the signal attaches to this stimulated G protein going to activate adenylyl cyclase, this one going to continue? No. There's a regulation. The cAMP levels are not going to always be up. So they are not always going to be up. So there's an enzyme which we call phosphodiesterase. Phosphodiesterase enzyme, as the name implies, is going to break down the phosphodiester bond because the cAMP molecule has the phosphodiester bond. So it's going to break it down back into AMP. So that one will decrease the level of cAMP. So it will restore the system back to normal. Again, there are certain drugs or certain things that we take every day. For example, caffeine. Caffeine will, at high doses, will inhibit this enzyme. Cyclic adenosine monophosphate would not be converted to AMP. So again, caffeine inhibits the enzyme phosphodiesterase, meaning the cAMP levels will rise because cAMP levels cAMP will not be converted to AMP. High amount of cAMP means high amount of excitation. That's why you'll be more active with caffeine.